I will talk about the underrepresentation of genomic data in cancer research and its consequences. I wanted to thank the STEM Quest team for giving me this opportunity to talk about this subject that is so dear to me. So I hope you will enjoy it, and I hope you will learn some things. So first of all, a little bit about me. I'm sorry, my computer is a bit slow, so I don't know, it's not going to the next slide. Sorry. So, oh, here, I'm sorry. So, a little bit of information about me. So I am a first-year master's student in biotechnology and bioinformatics at the Strasbourg School of Biotechnology in France. So I live in France. I have a bit of a research background in pharmaceutical innovation and cancer genomics. So I wrote my bachelor's thesis on how pharmaceutical companies can adapt their pharmaceutical and technological innovations to the needs of developing countries. I focus this on cancer genomics and on the work of Roche in sub-Saharan Africa. So this year, my research is more focused on the biology side of it, so on breast cancer and population genetics, whereas last year it was more about the finance, the strategic partnerships, and the technology and informatics behind all of these initiatives. So first of all, I would like to start by asking you a question. So when you hear or when you read that a new cancer treatment has been proven to work, what does this mean for you? So you can log into slido.com with this code or scan the QR code. And if when you have done this, please give me a thumbs up so I can activate the poll. Okay. Yeah, that's because I haven't started it yet, but I will start it right now. And you can answer, you can just say what goes through your head when you read this kind of information on the media, and then we can analyze results together and kind of talk about it. And to see some answers, really good answers. So hope for them and good luck. Hope, new hope for a life without the burden from cancer, innovative interventions, effective treatment has been shown to work in a patient population. Very good. Okay. So these are really good answers. I'm gonna wait for some more answers. A clinical trial was conducted to confirm whether this treatment is active or effective against the disease. Really good. So thank you for all your answers. They're really interesting and really hopeful too. So here is a bit of an answer about what the sentence really means scientifically. So, great. So what proven to work really means it's a reference group. We've seen in the answers that some of you gave, the drug successfully killed in a specific group of people who participated in the clinical trial. So this means that the treatment was effective for the population that was tested. So it means we have found a specific biological switch or biomarker, which I will explain later, that can stop a certain type of cancer. And it proves that we can use genetics to design targeted, smart drugs rather than just using general chemotherapy. This treatment will be used as the new benchmark. This is really good. This is a sign of hope for people who have the same type of cancer, but they also have to have the same genetic information, the same DNA. They have to be part of the same cohort. And this is great. This is scientific breakthrough and innovation, but not for everyone. So the problem is that this also means that the proof is incomplete because biological triggers for cancer vary by ancestry. A drug designed for one population's mutation might miss the target entirely in another. The big problem that we have nowadays is that only most of the information about DNA and the genome that we have is based on European and North American populations. And DNA really, really varies across populations, even across populations from the same continent, from the same region. So it becomes a problem with when most of the information we have is based on one population, and we're trying to treat people from other regions of the world with that same treatment. For example, a patient with lung cancer in Ethiopia will be treated with the same treatment as a patient with lung cancer in France, but it has nothing to do nothing to do with each other most of the time because the mutation is not the same, the reason for the cancer is not the same, and here comes the bias. So a proven dose can be highly effective for one group, but toxic for an underrepresented group because of differences in DNA. The dose that we will give to a European individual will be the same that we will give to someone that lives in North Africa, for example, because it's the only proven dose that we have that works, that we know works, but we only know it works for the individual living in Europe, not in North Africa. So this is when treatment becomes really biased, because, as I said in the slide, if the research only looks at a standard map, which is made of 80% European DNA, we have created a cure that we treat as a cure for everyone because of the lack of information. So now we will talk about what cancer really is. So each cell contains DNA, so it's your biological instruction manual. Normally, the cell follows a strict biological control system called the cell cycle. So the system checks if DNA is copied correctly, if the cell is healthy, and if it should divide, or if it should self-destruct, which is apoptosis. So there are systems in our body in place that recognize cells that have been mutated, that are not that are so yeah, so they have mutations, they are not I'm sorry, I'm struggling to find the words, but I think you've understood what I wanted to say. So they are not what they're supposed to be, and we have a lot of mutations going on in our bodies all of the time, and we have specific cells and systems in place that kill these cells so that they don't become so that they don't stay and provoke more mutations in our body. The problem is that cancer has a way of mutate the control genes, and then the genes will stop responding to biological stop signs. They will continue dividing, they will avoid apoptosis, which is cell death, and they will accumulate mutation. Cancer does this by downregulating what makes our immune system recognize them. So a cancer cell will have something in the surface that is not recognized as self, so that the body will know is not supposed to be there, so it has to kill it. But cancer cells and mutated cells have a way of downregulating that kind of flag that they have on the cells. So our immune system doesn't recognize them. They keep on mutating, they keep on developing. And as we can see in this picture, we have a chunk of normal cells that can become abnormal cells. Now, normally, if our immune system is healthy and recognizes these cells, and if the cancer cell hasn't had the time to downregulate what makes the immune system recognize it, it will just like be 2 or 3 abnormal cells that will be then killed or shut off. But if these cells downregulate what makes our immune system recognize them, they will multiply, and they will create a tumor. So it will become a malignant cancer that then grows its own blood vessels and invades the surrounding tissue. And so this is what makes cancer dangerous, because it will develop its own blood vessels. It will steal energy from other parts of our body that need it to feed the tumor and to make it grow. And they will also invade other tissues. So I would like you to go back to Slido and answer this question. It's just to know if you know the term or not. Here. So what is metastasis? So it and hinted it in the other slide, but I didn't really say what it was. So maybe we can go back and answer and see together what this means. So I will stop the other poll and start this one. Oh. Do you see the questions? I feel like there has been an error in this question. Okay. Okay, good. If it's a quiz, then you can answer, and we can see. Now you see the question. Yeah, no. Great. So most of you have responded, have the correct answer. So yes, metastasis is the spread of cancer cells from a primary site to form new tumors in distant parts of the body. So this is really the critic stage of cancer, because it leaves its primary site and it goes to it spreads in all of our other organs, which makes it so much harder to treat it and to target all the cancer cells, because it literally spreads. So um, now on to the next slide. So I have talked about kind of what cancer is, but now to you about DNA and how cancer affects our DNA. So most of you may know this, but so DNA is a molecule shaped in a twisted ladder. So it's the double helix. It has 4 chemical letters, so nucleotides, A, T, C, G for adenine, cytosine, guanine, and thymidine. And these genes are instructions to build proteins. So these genes are located, are part of the genome, which are in the chromosomes in our cells. And proteins are molecular machines that build the cell, that repair DNA, that control growth, and that trigger death programs. So DNA controls what your cell is allowed to do. It gives the instructions of what proteins should be produced and how these proteins should act in our body. So as we can see here in the picture, so adenine always links to thymine, and cytosine always links to guanine. I have another picture to explain kind of the double helix and chromosome image. So here you can see that, as I said, adenine teams up with thymidine or links, and guanine teams with cytosine. This is the double helix, and a part of this double helix is a gene. Then the double helix rolls itself around nucleosomes, which then form a chromosome, which are stored in the nucleus of all of our cells. So these are the literal instructions of how our body should work. And what's specific about everyone's DNA and genome is the order of these letters. So the order of all of these letters give the instructions to build our proteins. So the DNA, as I said, is stored in the nucleus. It doesn't leave the nucleus. What leaves the nucleus is RNA. So it's a copy of our DNA that can leave the nucleus. So DNA stays protected inside the nucleus, and when a gene needs to be used, the cell will make a copy called RNA. RNA will travel to the ribosomes, which are the protein factories, and the ribosome will read RNA and build proteins. And this is called the central dogma of molecular biology. Some of you may know it, I'm sure. But it's just a little recap to show you how this affects cancer, because if there's a mutation in our DNA, so our instructions are faulty, this faulty copy will be made into RNA, sorry, and then proteins will be built based on these RNAs, and then the mutation will just go on. So, as I said, so cancer is just genetic reprogramming. It's, oops, sorry, I forgot there was a question here. So this is kind of obvious, but what do you think happens if instructions contain errors? This is another quiz in Slido. So if you can re-log, and I'll start the poll. So here I'm talking about the AI because nowadays AI is what diagnoses cancer. Yeah, I have some answers here. Okay, so the right answer, as most of you have voted, is that the AI will produce false positives and less accurate health predictions for underrepresented groups. So if the instructions we gave to AI are faulty, then all that it knows how to do is act on the information it already has. So it will produce false positives and less accurate health predictions for underrepresented populations. Great job. So let's dive into our next part. So just to continue on what I was saying, so cancer is genetic reprogramming. I've explained all of this to you just to show you that the mutations that change in the DNA will have an impact on the RNA, and the RNA will create abnormal proteins. And this is what cancer does. Those proteins will force continuous division, will shut down repair systems, and will activate invasion pathways and silence apoptosis. So this is one of the proteins that is kind of seen as the genome guardian. So this protein recognizes DNA damage, and it produces a protein that pauses division, repairs DNA, or kills the malignant cell. So this is a very important protein that we have. It's called p53, and when expressed at normal levels, it recruits and regulates several processes that regulate oncogenesis, cell cycle arrest or progression, apoptosis, and oxidative stress. TP53 is activated both by endogenous or exogenous signals and prevents cancer cells from dysregulating metabolic pathways, multiplying, and invading other healthy cells. So this is really the protein that is keeping us safe from all the mutations that happen in our body. So most cancers in Europe or in North America, they just bypass. So they kind of know how to bypass this protein. So they hide, as I said. They stop showing what makes them a cancer cell. And so the protein will not recognize them. It will not recognize them, which helps them multiply and invade other healthy tissues. The problem is, so this is already a problem in itself, but the real problem is when TP53 is, so P53 is mutated. If there's a mutation in this protein, it's not only that it doesn't work and it doesn't recognize cancerous cells, it's that it helps cancerous cells develop and attack healthy tissues and steal energy from other metabolic pathways and nutrients, it really makes it worse and a lot more aggressive, because it's not just not recognizing, it's helping cancer. And the thing is, in many African tumors, this TP53 protein is mutated. It removes the main safety brake, and it becomes genetically unstable and aggressive. And I found in my research that a lot of African populations have a predisposition of this protein being mutated. So there's a little bit of an error in the genome that makes it really possible to be mutated if if mutations happen. And this mutation is not widely known or studied in Europe or in North America, because most of cancers are recognized with markers. We'll explain later. So we can see how this is a real problem, because if we try to treat a woman for breast cancer, for example, in an African country, and we're not looking for this mutation, it completely changes the treatment, and most of the time we don't even diagnose this woman or this individual accurately, because we're looking for biomarkers of other populations. We're looking for expressions of cancer from other populations. We're not looking for a mutation in the TP53 because it's just not known. So this is where I will explain now the biomarkers and what it is, and why it's so important to have an accurate map of biomarkers of every population. So what biomarkers really is a measurable molecular signal that represents a biological mechanism inside the tumor. It's like a window into how the cancer cell is wired. And inside every cancer cell, there are thousands of molecular pathways and networks of proteins that control growth, DNA repair, energy use, immune evasion, and survival. So a biomarker identifies which of these pathways the tumor depends on to stay alive, because every tumor is different and every tumor depends on something keeping it alive. It's stealing something, it's stealing a hormone, a protein, nutrients. It's stealing something to stay alive from somewhere else. But if we don't know what we have to look for to starve this the cancer cells, we can't treat it. For example, during my research in breast cancer, I've of course found a lot of molecular biomarkers. The principal molecular biomarkers for cancers in Europe are ER, so estrogen receptor based cancer. So this is all for breast cancer, and ER cancer feeds off of the hormone estrogen, which are found in females, and this is what gives it the energy to grow. This is what gives it the energy to downregulate it, to survive, and to repair its DNA. And so I'm sorry, I lost my my track of thought. So this specific type of cancer feeds off this hormone estrogen, and it's diagnosed when the estrogen levels are too high in a female individual. So this is what we look for first in Europe, right? We see if the estrogen levels are too high. If they are, and we diagnose breast cancer, then it will be treated with hormonal therapy, because the cancer is feeding off this hormone to grow. There's also HER2, the HER2 biomarker. So the HER2 is kind of like a growth signal. So this is a cell that will tell other cells to multiply. So it will tell them, now it's a good time to multiply. And cancer will use this system to shut down other, for example, I'm sorry. So the cancer will use this to amplify the signal to overdevelop in some sort. So this is a very normal cell that will just tell other cells to multiply. And the cancer cell will use this cell to tell other cancerous cells to over multiply it when they shouldn't. So normally other repair systems will come to shut down these cells, but because cancer uses this pathway, it will just lead to a very, very fast development of it, and immune cells will not be able to recognize that this is a cancerous cell because it's developing so fast. So for this kind of cancer, we use immunotherapy because the immune cells need to recognize it and to shut it down. And there's another cancer, there's another biomarker that's very widely known in Africa right now, thanks to late studies, but wasn't very known a couple of years ago. So it's BRCA1. So it's a gene that it's a gene repair mechanism. So if there's a DNA mutation or something is not well, it's not going well in the DNA, this gene will just repair it and send signals to the body that this specific gene needs to be repaired. But cancer, once again, will shut down this gene and will use it to its advantage. So this one needs chemotherapy because it it involves a whole gene. So these are just 3 biomarkers that exist in breast cancer. There are a lot more, but you can see how 3 biomarkers need different therapies. Each one needs different therapies, and each one attacks something different in the body. So when, for example, a woman in Asia goes to the doctor for, I don't know, microcalcification on her breast, or something's not feeling right, because they only have information about European and North American countries, they will look first and foremost for ER, so estrogen receptors, and PR, which are progesterone receptors, which is also a hormone that cancer feeds off of. So if the levels are too high, we know that it's breast cancer. But if they're normal, we will just send this patient home telling her, okay, so your hormone levels were normal. This must be a benign tumor or maybe a cyst, but it's not cancer, when it actually is. It's just that we didn't look for the right things. So this goes a bit with what I just said. So how underrepresentation breaks medicine. Modern cancer medicine is built on genomic datasets, and these datasets mathematically define biomarkers, drug targets, and AI diagnostics. If most data comes from one population, biomarkers describe only that tumor biology. Drugs are optimized for those molecular pathways, and AI systems learn only those tumor patterns, so they will not look for anything else because your information is not in the system. And this is very dangerous. If we cannot diagnose you because your information is not in the system, it's like we're letting you, we're literally letting the disease get worse and worse and worse until there's no turning back, there's no hope for treatment. So other populations are treated using a foreign biological map, which is very problematic. So I'm sorry, this low. So here, once again, we're going to talk about the African example. So in a European-based model, biomarkers are defined from European genomic patterns. The drugs are optimized for those molecular targets. AI diagnostics are trained on European tumor images, and there's a higher response to treatment, slower progression, and earlier diagnosis. Now, the thing is that the problem is not just these. I mean, the big problem is that the genomic map that exists is only based on, it's mostly based on the European-based model. And the thing is, big pharma companies will not try to adapt their treatments to other, or their research to other populations. This, for example, in the African tumor biology example, so there are different mutation landscapes. Oops, I'm sorry. So there are different landscapes like TP53, as I've explained before, or DNA repair genes. The key drivers are often not targeted. There is lower detection accuracy, lower response to treatment, faster progression, and later diagnosis. Patients are treated using a foreign biological map, using information that doesn't even, it has nothing to do with them. So this is scientifically incorrect medicine. If your DNA is not in the databases, or the DNA of people who are genetically like you is not in these databases, your survival probability is mathematically underestimated. So every new medical AI system is trained on genomic and pathology data. And if your DNA is missing, AI will learn to misdiagnose you. And future medicine will literally become algorithmically discriminatory. So I have focused my work, and I have done an internship in Roche, learning how this really works. And Roche is a really big biotechnology company that I think most of you know, and they have trying to take on more projects that will help the greater good and will not focus only on profit. So Roche started sequencing African populations and rebuilding biomarker panels. So sequencing is kind of reading the code of life. So it's literally reading the DNA, the genome of every single person. DNA sequencing is a laboratory process of determining the exact order of the 4 chemical building blocks that we've talked about before, called bases, that make up a DNA molecule. So by doing this, Roche found new mutation clusters, new immune signatures, new drug targets, which means new medicines become possible, which means we finally know what to treat in these populations. By actively sequencing African populations and rebuilding biomarker panels, they're ensuring that the biological maps used to train these AI systems actually include the global majority. So we've talked about what happens in our body, but we can also talk about the treatments that exist and why, once again, the genomic underrepresentation of certain populations are detrimental for their treatment. So we will talk about monoclonal antibodies. So monoclonal antibodies are engineered immune molecules designed to seek and destroy. They're programmed to ignore healthy cells, and lock onto a specific molecular flag on the surface of a tumor known as a biomarker, which we've talked about before. So for these drugs to work, the antibody must fit the biomarker perfectly. And once attached, the drug can block growth signals or mark the cell for the immune system to kill. So as we've seen before, one of the biggest problems in cancer is that our immune system does not recognize them. Our immune system doesn't know that this is not a normal healthy cell and it should be destroyed, which allows it to develop however it wants at the rate it wants. And it's not really threatened by anything in our body because our body does not recognize it as harming, as something that's harming for us. So monoclonal antibodies are kind of these flags that will attach. So here we can see the antigen binding site. This is genetically modified in the lab to fit perfectly the expression of your cancer cell. So of the cancer cell and the type of cancer. So antigens will dock into they will dock to the cancer cell, and this is kind of a flag that shows our immune system, look, this is not normal. We have to kill it. You have to kill it. You have to get rid of it. Or it can block the growth signals by itself, but it's more of a tag. I see a comment. I think it's also used in enzyme replacement therapy to allow the enzymes to cross the blood-brain barrier. Yes, excellent remark. It's also used. Monoclonal antibodies are used in a wider variety of subjects but yeah, so if the biomarker was discovered and mapped using mostly European DNA, the drug is built to recognize that, and if an African patient, for example, an African patient's tumor expresses a slightly different version of that biomarker due to genetic variation, the antibody cannot dock. So if you send a monoclonal antibody made in Europe for a specific type of cancer into a patient in South Africa or, I don't know, Vietnam, this is not guaranteed to work. Cancer treatment is never guaranteed to work, but this really lowers the chance of the patients, because we do not know what the antibody needs specifically to dock on. Whoops, I'm sorry, I think I just closed the page, but I don't think. I'm really sorry. My computer just closed my slides, but I think you still see them. Okay, great, you still see them. So yeah, these are what monoclonal antibodies are used for, and this is why, once again, if we don't have the data, if we don't know what the cancer cell looks like in a specific population, the treatment it can be defined as useless. And it's a shame, because monoclonal antibodies are very expensive treatments. They take a long time to make in the lab, and if it's something that is not going to help the patient, it's just a shame. So here's a next question. So another question, just general knowledge. So do you know which of the following is the most well-known monoclonal antibody used to treat HR2 positive breast cancer? You're not supposed to know this. I haven't said it. It's just general knowledge to know if you've heard it. So let me start the poll. Normally, oh no. Normally you can see it now. So I see some answers. Most of you have answered trastuzumab, which is the correct answer. Trastuzumab Herceptin is used to treat HER2 positive breast cancer. And so a little fun fact about monoclonal antibodies, and that can help you recognize the disease it's targeting if you see it, somewhere, is that so all monoclonal antibodies end with MAB, which means monoclonal antibody. The beginning of every monoclonal antibody name doesn't really have significance. It's just to differentiate different monoclonal antibodies from each other. And where this really becomes interesting is that what really holds meaning, it's what comes between these two. So between MAB and the 3 or 4 letters that are there to differentiate monoclonal antibodies. So for example, we have trust, that doesn't really mean anything. Two, which means that this monoclonal antibody is made to target tumors. And zoo. So zoo means I I I I I think it means that it comes. So the treatment comes from mice stem cells or chimeric stem cells, which are stem cells that are cross mice, cross human, and yeah, and then you can just really recognize what the treatment is targeting. So LE is immune system, and in the other is 2, which is tumor, and XI is actually chimeric, so part human cells, part mice stem cells. So yeah, and there's a lot more of other little words that define what the monoclonal antibody is made for. So thank you so much for answering. And now, so we'll talk about the medical consequences, which I've talked about a little before. But so the drug will miss its target or will attach too quickly to be effective, but it will not attach to to what it's supposed to attach to. I'm sorry. The cancer will continue to grow and spread despite the expensive treatment, and the precision medicine becomes imprecise. I'm sorry, leading to poorer outcomes for underrepresented groups. So genomic diversity in research isn't just about equity, it's a functional requirement. We cannot say we have made scientific breakthrough and that we have found, treatments that are just unilaterally benefiting one population. Of course, it's great that we find treatment for European and North American populations. It's really important, but it's just as important to diversify and to look for other genomic signatures and ways to include these other populations also suffering. So without inclusive data, billion-dollar therapies remain biologically blind to a massive portion of the global population. So to conclude, cancer does not discriminate, yet our data creates a divide. A smart missile, so a monoclonal antibody, that it's supposed to directly identify the target, but because it's genetically modified to find it, if it cannot find its target, it's not precision, it's a failure of data. And we must move beyond the European reference genome to see the full picture of human health. So inclusion is the only path to correct science, and correct science is the only thing that saves lives. Thank you so much for listening. Thank you so much for answering the little Slido question and for your attention. And yeah, thank you so much. If you have any questions, please let me know. And I hope you've enjoyed and I hope you've learned some things today. If you have any questions, please put them in the chat or yeah, feel free to ask me anything. Thank you so much.
All right, guys, if you have any questions, please do put them in the chat box. Otherwise, we can conclude the meeting. We'll wait for a minute or two to see if we have any questions, and then we can conclude the meeting.
Thank you so much. I'm really glad you found it insightful, and I really hope you've enjoyed it and learned some new things. And also, I think it's really important to sensitize. I think I'm sorry, English is not my first language, I think, but yeah, to really spread the word about why this is a problem and why we can't just leave other populations on the side. So yeah, thank you so much. It's really nice of you. Thank you.
All right, seeing as there are no questions, thank you so, so much, Yasmin, for the presentation. Oh, it looks like we do have a question.
Okay, so this also includes the drug they prescribe for the disease only worsens and not helps them, right? So yes, it can either just not have any effect on them at all, or it can, you know, because the monoclonal antibodies are made to be efficient, so they have really high affinity. They need to dock into a cell, so it could dock into a healthy cell that has, the same structure that for example, the European patients that have this molecular structure, and it can just bind and kill healthy cells. So yes, this can worsen them. This can inhibit and neutralize healthy cells for the individual because it's not targeted enough. So I hope this answers your question. And how come the body and immune system don't recognize cancer? Okay, so this is a really great question too. So the cells have something that is called MHC. So MHC is kind of let's say a polypeptide chain that is on their surface. So we can see so every cell has like kind of a flag in its surface telling the immune system, I am a healthy cell. I am a cell that belongs to the body, and there's nothing to look for here. Cancer shows a very so cancer cells who don't downregulate their MHC are very visible to the immune system. The immune system immediately recognizes them and kills them. But when cancer, because all mutations are different, some mutations are more efficient for cancer than others. Some cancer cells have learned how to downregulate their MHC. They have learned how they literally are playing a role. We could say they um, they look like healthy cells, so they have learned how to look like healthy cells, how to suppress what makes them look like a cancerous cell, like that when, for example, T lymphocytes or phagocytes, macrophage, natural killers go around the cell, they don't have any flag that tells them this is not good, this is something we have to kill. Cancer has learned how to downregulate it and how to come across as a healthy cell. So yeah, that's how the body and the immune system don't recognize cancer. And the treatment is not standardized. Is each patient treated according to their genes? So there are kind of two ways. I will say that I haven't dived in that much in my research yet, but there are monoclonal antibodies. So most of monoclonal antibodies are made from the patient's cancer. So we take a sample of their tumor and a sample from their immune cells. And then we do some work in the lab to really analyze how the cancer can be recognizable from monoclonal antibodies, and we genetically make monoclonal antibodies. There are other monoclonal antibodies that are more standardized and they are made in big corporations, and this is based on information that is known, because there's a lot of cancers that look all the same, so in Europe we have a database of all of the cancer cells in, for example, bone cancer look like this, so we will try this standardized treatment first because it's cheaper. It's still really expensive, don't get me wrong, but it's cheaper than genetically modifying and making a monoclonal antibody that is just made for you. So that's the first thing that we will use. It's a monoclonal antibody based on what has already been proven to work, because there's a lot of cancers that are just like phase 1 cancers that are not as aggressive, so we can directly kill them with monoclonal antibodies that work for other people because it's not as specific yet. That's why I'm pointing out this problem of underrepresentation is because if we had a monoclonal antibody, so a general monoclonal antibody that can directly kill a cancer cell in the first stages of cancer that is based on African genetics, this could lead to a lower mortality for this population because we could, catch the cancer at its early stages and send in a monoclonal antibody that will directly inhibit and neutralize it. So yeah, I hope this answered your question. So there are standardized and non-standardized monoclonal antibodies. Some of them are standardized based on cancers that are really general and especially in the beginning, and some are more so they're genetically modified and made for your type of cancer when it's already very aggressive and kind of yeah, in the further stages of cancer. I hope this answered your question. And I've learned that in immunology, it's connecting MHC, exactly, antigen receptors and macrophages and helper T cell is such an, yeah, exactly. You've said everything. So yeah, it helps the macrophages and helper T cells, which kill cells that are not recognized as self and cells that are not good for us. And it connects it and shows them, look, this is not good for you, for us, you should kill it. And so yeah, that's immunology. I would love to go on about that subject too because it's I'm really passionate about that. But yeah, so thank you so much for your questions. They're really interesting. Are
All
there
right. Seeing as there are no more questions, I believe we can conclude the meeting. Thank you so much, Yasmin, for the presentation. It was truly very insightful. I'm sure everyone here learned a lot, including myself, actually.
oh,
And
great.
thank you for being a part of STEM Quest.
Thank you
With
so
that
much.
being said, we can conclude the meeting.